So today's lesson, like I said, we're looking at evolving pathogens. So essentially, with humans or with any living thing that we've looked at, we look at how changes in their allele frequencies can lead to them evolving new traits and eventually even diverging as a new species to become a different population. With bacteria, this is supercharged for two reasons. One, the growing time for bacteria is much more shorter, right? They can reproduce at a rate that is insanely fast compared to any other living thing because that's how small they are and that's how quickly they can reproduce. And also another thing is that due to the fact that they can reproduce in such high numbers, they can also accrue mutations much easily because they have the sheer numbers, right? If we looked at all the humans on Earth, we would see there's a lot of variation, a lot of differences, in the same way that if you look at one colony of bacteria, there's millions of them there. So there's going to be millions of variations that allow them to be basically selected for. So today we're going to look at how these pathogens have evolved as a result of this. And let's get started. All right. First, let's do the question. Cows are the main source of milk in Europe. Dairy cows breeds can produce 25 liters of milk each day, more than their wild ancestors. So the first original species of cows could not produce 25 liters of milk, but today, in the age that we live, cows are able to produce that much milk with each day. Describe how an increase in production of cow's milk could be achieved by farmers over many generations of cows. So what is allowing for them to increase their production in the milk over the generations by the farmers? Who can I ask? Emelinda, what do you think? What? Selective breeding. So could you shape that into an answer? How about Yasmin? How would you give me an answer for this question? So put it into the context of the cow milk production. So in a question like this, I know the straightforward answer is just saying selective breeding, but they are giving you 2 marks for this question. So you must give a worthwhile explanation. So what you should say is, over many generations, cow breeders have selected for the advantageous phenotype of an increased milk production, thus allowing for the trait to be passed on generation after generation, developing the trait, giving it to a point where cows could produce up to 25 liters. Okay, so there are a few things you should say. You definitely need to mention selective breeding, and you also definitely need to say that the trait of high milk production was artificially selected for, and you have to mention the four principles of natural selection, which are variation. Second is selection pressure. For here, it will be an artificial selection pressure. Third is the advantageous selection of that phenotype. And then fourth is the heritability. So it's passed on. You have to describe it based on those 4 principles, and that's how you would get the full marks for that, right? Or you could just say cows would select for cows that produce milk, and the cows would be bred. That is basically a short answer. All right. So today's lesson, we're looking at what is a pathogen and how pathogens have evolved to be able to gain gain abilities such as bacterial resistance, and also how something like antigenic drift and antigenic shift has allowed viruses to also evolve as a result. So we're looking at 2 specific breeds of pathogens. One is bacteria and the other is viruses. The other ones we're not going to look at very deeply today. Right? So glossary task. So what is a pathogen? Simply put, a pathogen is a microorganism that is able to cause a disease, right? It doesn't have to be something specific. It could be a virus, it could be a fungus, it could be a bacteria, or any other microorganism. As long as they are able to cause a disease, they are considered a pathogen. All right. So any microorganisms that you have inside you right now in your gut, they aren't considered pathogens because they are not causing a disease right now, but they can potentially become a pathogen if they suddenly cause a disease to you, right? So what are the two types of pathogens that you guys can look at? One is a cellular pathogen, which means a pathogen that has a cellular structure and actually is considered to be alive. Okay, these are your fungi, these are your bacteria, your parasites, and also protozoa. We also have non-cellular pathogens, which are pathogens that are not considered to be alive, which are your viruses and prions. Okay, viruses, whether they are alive or not, is a very hotly debated topic. But as of now, people usually consider them as not being alive because they cannot carry out cellular functions or do not have a cellular uh, do not carry out cellular functions on a regular basis unless they have infected a host. Then they can carry out functions. Otherwise, they become very innate. They don't really carry out any functions. Yes, so by itself, viruses do not reproduce. They can only ever reproduce in the host cell, right? That itself is one of those things that is important to be considered alive. Okay, so when we're thinking about whether they're alive or not, you actually go back to your year 7 science of thinking of what do you need to have to be considered alive. You need to be able to reproduce. You need to be able to consume food and release waste products. And when you think about all those things, bacteria, they're able to do that and they are considered to be alive. Whereas viruses, they do not actively do that. And with viruses, they do some of those functions, but they need to first infect a host cell and take over its functions to be able to do that. So it's still debated whether we would even consider them as being alive. All right. So we have those pathogens, and how we deal with those pathogens is by using something known as a antimicrobial agent. So these are agents that are able to kill or slow down the growth of these microorganisms, and they can come from any amount of sources, right? So they can come from basically all these different sources in different ways, right? For example, for bacteria, you guys famously know what agent we use is antibiotics. And one of the early sources for antibiotics was actually molds, right? That's how we discovered antibiotics in the first place, is by growing, plating molds, and then noticing that there was a difference in the growth of bacteria around the molds, showing that there were uh, basically bald patches around the mold because of the fact that the mold was producing a chemical known as penicillin, which was a first antibiotic that was discovered, and that is what allowed us to make our own as well. So each of these acts as a source of antimicrobial agent, and each of those antimicrobial agents can have different ways of working, right? For example, how it works, the chemical structure acts to basically kill or slow down the growth of pathogens, or it can target different things, right? Targeted pathogens, these different antimicrobial agents basically target different pathogens specifically, right? Antibacterials obviously work on bacteria. Antivirals basically only work on viruses. Antifungals basically only work on funguses. So some of these antimicrobial agents are very specific to their specific pathogen, while some can be very general as well. For example, something like a disinfectant or an antiseptic is more of a broad spectrum antimicrobial agent. You guys don't need to know all these different terms. What you just need to know is that antimicrobial agent is something that kills or slows down the growth of microorganisms, and they can come in many shapes and forms, and they can be found all around us, right? If that is microbial agent is, antimicrobial resistance is considered as the ability of the microorganism by exposure to that antimicrobial agent. So resistance is able to basically survive being exposed to it. And one of the most famous ones is antibiotic resistance, right? So what is an antibiotic? They're basically the medicine that you can use to treat any disease caused by bacteria. Not all bacteria and not all antibiotics work for all bacteria. In fact, one of the most common antibiotics that was used early on, penicillin, is actually considered to be non-functional against bacteria, against the bacteria of today because they were overused. So they're no longer as functional, not as useful nowadays considered to back then, right? Some bacteria would have become resistant to these antibiotics, and we're going to look at how they can become resistant today. But essentially, how can these antibiotics work? There can be multiple ways that they actually disrupt the function of the bacteria. Let's look at them. So bacteria that are resistant to bacteria that are antibiotics, how did they gain this trait of antibiotic resistance? So it's obviously through natural selection, like any other trait that is gained. Any advantageous trait adaptations that are gained are usually going to happen through natural selection, which is going to select for that trait, right? So what happens? We have a colony of bacteria, harmful bacteria. That in that colony, there exists a natural variation of traits, right? Caused by mutations within the bacterial DNA. Suddenly, one trait will emerge that allows them to become resistant to antibiotics. And what happens next is the selection pressure. What happens if these bacteria are suddenly exposed to a high concentration of antibiotics? They will suddenly die off. But what happens is natural selection will select for those bacteria that are resistant against the antibiotic. And as a result of selecting for it, they will have a selective advantage in that population. And what's the last one is heritability, whether they can pass on that trait to their offspring. And as you can see, that's what happens as soon as those bacteria that are resistant to the antibiotics survive. They will pass on the trait of resistance to the next generation, and that generation will pass it on to the next generation, and so forth, right? So the 4 principles of natural selection are at work here, and you can always track them down when we're talking about natural selection. Okay, we start off with natural variation in a population. Second is the selection pressure, and here the selection pressure is the antibiotics. The third one is the selective advantage or the phenotype, and of course, the bacteria that are resistant to antibiotics have the selective advantage. And lastly is the heritability, which is where the bacteria can even exchange genetic material through a process known as bacterial conjugation. Don't know why, but my throat is pretty dry. All right. So bacteria don't only just reproduce to share their genetic information, they can even share genetic information between each other, known through a process known as bacterial conjugation. That's basically, think of it like when one bacteria dies or is able to make contact with another bacteria, they can basically share their genetic information with each other by passing on the advantageous genetic information, right? So again, let's have a look here. Follow through, right? One is the variation. A population of bacteria has individuals resistant to an antibiotic as well as individuals susceptible to the antibiotic. So the reason why the antibiotic resistant strain isn't proliferating here is that there hasn't been a selection pressure yet. So they only have that variation existing here. When there is a selection pressure that selects for that trait appears, suddenly the rest of the population is wiped out, and the bacteria that are able to be resistant to the antibiotics survive, and they have a selective advantage, allowing them to confer that trait into their next generation. And obviously, the bacteria that are resistant to the antibiotic are able to continue replicating and pass on that advantageous allele of resistance to other bacteria through the process of bacterial conjugation as well. Okay, so there's two ways bacteria can pass on that genetic information. One is through reproduction, that is the old-fashioned way. The second way is bacterial conjugation, which is basically a method of them sharing genetic information between each other. It's basically like me coming to you and saying, would you like my advantageous phenotype of being tall? And I somehow pass on that genetic information to you. Of course, with humans or any other organism, it's not possible. But with bacteria, they're able to actually share that genetic information across one another. Okay. So we've talked about how they can be resistant, but we should also talk about how they can suddenly emerge a population of resistance to antibiotics. So let's talk about how that can manifest itself, right? What does it mean for a bacteria to be antibiotic resistant? So resistance to antibiotics can manifest in a few ways. There's not just one trait that can cause them to become antibiotic resistant. There are a few possible traits that they can obtain to become resistant against bacteria. Because resistant against antibiotics, because antibiotics act to suppress the growth of the bacteria in a few different ways. So the bacteria can become resistant against the antibiotics by doing a few things. The first thing it could have done is basically making sure those antibiotics cannot even get into the cell, which is to change the cell wall of the bacteria to become more resistant against antibiotics, basically impermeable towards antibiotics. The second thing it can do is to change the shape of the protein targeted by antibiotics. Basically, these antibiotics want to target bacteria. So once bacteria is able to modify that specific protein to become non-conformational towards the antibiotic, it won't let the antibiotic bind. Thus, the antibiotic won't be able to function. Remember that proteins are very shape specific. So if the original protein that allows the antibiotic won't be able to bind to that protein and won't be able to deactivate it. So by changing the shape of that protein that is being targeted by the antibiotic, bacteria can even survive the antibiotics. Third one is increasing active efflux of the drug. Basically, being able to pump out the antibiotics as they enter the bacteria. That is also another way that bacteria can become resistant against antibiotics. And lastly, is inactivation of the antibiotics, which is to add a phosphate group to the antibiotics to reduce the efficiency of the antibiotic itself. All right. So this happens because by binding a phosphate group to the antibiotic, it reduces its ability to bind to the ribosomes of the bacteria. So main four ways that antibiotic resistance can be obtained, each of them by evolving one or the other, they can suddenly become resistant towards the antibiotics. All right, any questions about this? All right, so now that you guys have had an idea of how antibiotic resistance can be obtained, let's have a set of what that means. Well, super.
Except that it's 2 feet by 4 feet. Now, the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, of course, the thin agar, that bacteria can move around it. The background is black because it's ink in it. The bacteria appear as white. First, you see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So let's go through this in the scope of natural selection.
Not too much antibiotic.
I have a think of what's happening here, right? Right. We start off with the wild type bacteria. Okay, these are the first strain of bacteria. They grow into the there's no antibiotics here, so they can grow uncontrollably here, right? So when they get to the boundary, that's when they get exposed to the plate that has antibiotics on it, and you realize that they can't grow on it at first. This is when natural selection happens, right? The variation within the population of bacteria here, there will be at least 1 or 2 of them that is resistant to this antibiotic. Suddenly they are able to reproduce and grow in this medium. And same, it happens again and again and again until they're able to survive at 1,000 times the concentration, the original antibiotic. So this is how bacteria suddenly can become resistant to antibiotics, is because they follow the principle of natural selection, right? There exist millions and millions of bacteria within a single population, and within that population, there will be variation of certain bacteria that can be resistant towards an antibiotic. And when there is antibiotics, that is acting as your selection pressure, selecting for that advantageous trait of resistance, and they will then pass on that advantageous phenotype to the next generation and so on. This is how they can actually become resistant to the population, to the antibiotics over time. So it's just a matter of time for bacteria because they have sheer numbers. And as we know with natural selection, if you have a lot of numbers, that means you have a lot of variation, and that is going to drive natural selection. Right? So why is antibiotic resistance one of the biggest socio-scientific issues in the modern age today? Okay, it is actually considered to be a very, very big danger for antibiotic, for not just the scientific field, but the entirety of the human race. Because before the invention of antibiotics, we were very much compromised whenever we got any bacterial infections. Okay, our abilities to combat bacterial infections were very small before the invention of antibiotics, because they were basically able to infect us at a very high rate because they are all around us, right? When we were able to discover antibiotics, we managed to find a way to combat them And today we are relying on them wholeheartedly because every process we do, every operation, every organ transplant, every process that requires the exposure of something to bacteria requires antibiotics to combat those bacteria so that they don't get compromised. And so we are using antibiotics at a unprecedented rate since their inception, and that is actually acting to be the problem, right? So why is it actually acting? Why is it considered to be one of the biggest problems? Antibiotic resistance is because how we use it is allowing for bacteria to suddenly become not suddenly, but become more and more resistant towards the antibiotics that we have. For example, the first thing is inappropriate compliance plan. Basically, ask yourself whenever you've been given antibiotics by a doctor, have you finished your course? Have you been the person that actually finishes all the antibiotics you take? If you haven't, you are contributing to the problem, right? Basically, incomplete course may not sufficiently eliminate all the pathogenic bacteria that is present in your body, right? You're sick, you start taking antibiotics, you start to feel better, so you decide, why do I need to finish them? I'll just stop it as it is. What can happen is that essentially even having one or two strains of bacteria that haven't been killed by the antibiotics can allow them to continue replicating within your body. And if you give them enough time, they will then become resistant to whatever antibiotic that you are taking, right? Suddenly you have a bacteria that is resistant against the antibiotic that you have, and it won't work anymore, right? That is one of the problems. The second thing is that next is, do you think doctors are so responsible about prescribing antibiotics? When antibiotics emerged as a miracle drug that can save people from all bacteria, doctors suddenly started to prescribe them like crazy, right? Anytime they saw you were sick, they decided, oh, it's a bacterial infection, you should just take antibiotics. Even when it was a viral infection. And if you haven't realized this, viral infections are caused by viruses and they don't get impacted by antibiotics. Antibiotics are specific towards bacteria and will only function against bacteria. For viruses, it's basically nothing, right? So if doctors keep prescribing them when there is no bacterial infection or no disease being caused by bacteria, what you're doing is that you are hurting the bacteria in your body that are helping you. Okay, remember that bacteria. There are some that are good within your body that help with your digestion and help with the maintenance of your gut. So by taking antibiotics unnecessarily, we are exposing our normal flora in our body to those antibiotics, and that can actually act to basically harm us in the long run. And overall, it can also develop resistance in the bacteria that can be found in your body, which is a problem because some bacteria in your body, they don't do anything at all. They don't harm you, neither are they advantageous. But what can happen is that if you get hurt inside, if there is bleeding, they can get exposed and make it become infected and actually cause you to get sick. And if they are already developed, have resistance, then you are worse off for that. Another one is the widespread use of antibiotics. Humans are not the only ones that receive antibiotics. In fact, if you put it into a chart of what organism uses the most antibiotics in the age today, it's going to be animals, specifically agricultural animals, right? Your farm, your cows, your chicken. When we are raising them, we want to make sure that they don't die off before they mature and we can kill them to eat them. So we feed them antibiotics to make sure that they don't get sick. But as we know, by overfeeding them antibiotics, we are basically allowing for resistant strains to appear within these animals. And we are eating these animals, so that increases the chance of us passing on resistant bacteria in our food to us, right? So increased use of these antibiotics can increase the probability that an individual prescribed antibiotic will be inhabited by antibiotic resistant bacteria, right? So specifically for agricultural use, the growth of animals for consumption is one of the most abundant uses of antibiotics. So they can suddenly emerge a population of emerge a strain of bacteria that is resistant for the antibiotics that are used for those animals. Okay. So anyone confused so far? Okay. Just remember, antibiotic resistance is a trait just like any other trait. It can be manifesting itself in 4 different ways. So there are 4 different ways that bacteria can become resistant to antibiotics, and bacteria can evolve, can have a mutation that gives any of these 4, then it becomes resistant towards the antibiotics, and it will be selected for through natural selection, be spread to the next generation, right? So that is for bacteria. Now let's look at viruses. Okay, we have also dealt with a lot of viruses that have been able to evade our basically evade being killed off because of the adaptation and modification that they go through, specifically at their surface antigens, right? So if you look at viruses, they usually look something like this, right? They look like a picture like this, where they have these structures on the surface. We call these surface antigens, okay? So what happens is that when the viruses basically undergo mutations at those surface, they can become less or more virulent, and also possibly even gain resistance to your immune system and become a more dangerous disease, right? A more dangerous virus. So the main reason that viruses can avoid detection by our memory cells in our body through vaccination or past infection is because they basically go through mutations that change their surface antigens. Basically, let me explain this in a simple way to understand. Imagine these surface antigens are how we recognize that virus, right? How your body recognizes this virus. If they change that surface antigen, can become unrecognizable towards our body's memory cells. So our body will think of it as a brand new virus, and sometimes even changing those surface antigens can make them more virulent, right? Basically, a more dangerous virus, right? Think of something like COVID, right? Think of all the different strains of COVID, right? We had the Omega strain, Delta strain, Alpha strain. Was that Alpha strain? I've already forgotten. That's how many strains that can exist for that same virus. Because what's happening is that is change the surface of those viruses. Thus, they can become different. So there's 2 main ways that this can happen. One is considered as the antigenic drift, and the other one is called as mutations. That's basically the natural process how mutations accrue, right? Small and gradual, and they can become somewhat unrecognizable. The second one is antigenic drift, which is a sudden and causing it to become almost recognizable to what it was originally, right? Let's look at those 2 examples. Okay, let's look at antigenic drift first. All right, this is when we have small gradual changes in the genes coding for the surface antigens. So this is following the steady rate of mutation, right? So that allows them to still be recognizable by the memory cells, because essentially, if there is antigenic drift happening, it is happening at a slow rate. So they are still almost the same, only small changes have been made to their surface antigens. So our memory cells can still recognize them. However, if this is allowed to continue for a longer period of time, enough mutated antigens will accrue allowing it to diverge from the original population and be considered a new subtype which can no longer be recognized by the memory cells in your body, right? So let me show you a picture of that. So yeah, essentially, at first this was the virus, and then it started to have mutations that changed its surface antigens. Okay, through those changes, it became somewhat unrecognizable to your memory cells, but they were still able to recognize it. But over many generations, the accumulation of those mutations allowed it to become unrecognizable to your memory cells, to your immune system, and becomes a new subtype of the original virus, right? So this is a slow and gradual process, but it can still become unrecognizable over time. However, the next one we're going to look at is antigenic shift. This is a very sudden and significant change in the genes encoding for the viral surface antigens. This is specifically going to happen in one situation when two or more strains of a virus combine when co-infecting the same host to form a completely new subtype. Remember that how do viruses replicate? They basically infect a host cell and make the host cell reproduce those viral cells, right? So if two or more strains of virus are co-infecting the same host, then they will both share genetic information and they will then create a new subtype through viral recombination. This is basically a cheat code that viruses can use to produce a brand new subtype almost instantly, right? So you can see here. Yeah, so there's virus A, virus B, both are different from each other. What they do, they both infected the same host cell at the same time, and they take over the protein machinery to reproduce. And as a result, both of them share that genetic information within this host cell, and those genetic informations recombinate through viral recombination to create a brand new subtype. So it is antigen drift. Right? So depends on which specific example you're looking at. If you're looking at something like I can't be able to relate it 100% because I don't know the information, but I would say it would probably be antigenic drift most of the time, but antigenic shift could have happened as well, right? For example, if you looked at the way they were classifying the COVID strains, sometimes you can say Delta V1, right? And then they can say V2. This indicates it is a antigenic drift, right? There's changes, but it's still considered under the Delta class as a version 2. But if we're looking at Delta, Alpha, and Omega, we could have gotten this either through the accrual of mutations creating a new subtype, or it could have been antigenic shift where there were suddenly recombination of different subtypes creating a new one, right? So I don't know the specific examples with COVID, but basically, let's look at this picture, right? So there's two examples here for a antigenic drift and an antigenic shift, right? So basically, we have the original strain here. It accrues changes in its surface antigen through mutations, and that is considered as antigenic drift, right? You can see how the surface antigens have changed. Another way it can happen is by infecting by basically infecting the same host cell and sharing their genetic information through viral recombination, producing a brand new subtype, right? And also another example. Yeah. So basically, if you're trying to differentiate drift versus shift, remember that drift would still maintain similar characteristics to the original wild type virus, right? So if this is the original influenza virus, through genetic antigenic drift, it would still retain a lot of the original structure of the virus, only have minor changes. But with antigenic shift, it would be much different due to it recombining with another virus subtype as well, right? So this is why viruses are also another dangerous pathogen, be considered as incredibly incredibly hard to treat because they can basically change their structure to become unrecognizable over many generations, right? So if you have ever wondered why every year we must get a flu vaccination, why every year we get a flu vaccination, it's because every year the viruses mutate and to become unrecognizable to the vaccine that we obtained in the previous year, right? So let's look at this in specific example, right? With when you're talking about your common cold, it is caused by a group of viruses known as rhinoviruses, which infect your upper respiratory tract. So due to the fact that because the high ability of the surface antigens to constantly mutate, they basically become unrecognizable to any vaccine that we take or any antivirals that we take. So it is extremely difficult to basically become immune to these new viruses because they are basically mutating to become a new virus every single year. So we need to constantly take a new vaccine or basically be able to combat these new viruses every single time it happens, right? So basically, if you look here, this is basically how it looks like, right? So we have a virus infecting our body. Our body is developing an immune response targeting that specific virus, and then that virus is taken care of. But over time, that virus somewhere around the world is becoming different through antigen change, and over time it's become unrecognizable and it has become a new strain that is infectious and it's infecting the population again, right? This will keep happening every year no matter what we try to do because of the fact that these viruses are constantly mutating and becoming new strains and becoming basically unrecognizable to whatever immune system that whatever immune response your body has created. Remember that your body creates memory cells, right? These memory cells are there to make sure that the same infection does not take hold in your body again. But if the viruses that are infecting your body are constantly changing, they are constantly mutating to become something else, then your body isn't going to be prepared for that. And this is most problematic with something known as influenza. So why is it that influenza, the seasonal flu, is still such a prominent and dangerous thing? Is because of the fact that they can have mutations occurring at their surface antigens to become unrecognizable, right? Be that through antigenic drift or antigenic shift. So with antigenic drift, there is changes that happen in the surface antigens, and this can either increase the effectiveness or decrease it, but basically the effectiveness of previously generated memory cells will be reduced due to those changes that occur at the surface antigens. And if enough changes can accrue, they can even become ineffective. Therefore, yearly vaccinations against influenza are required to be able to maintain the immunity. So what you think happens with your influenza vaccines every year is that basically scientists take research about what are the current influenza strains that are present in the wild type, and they have an idea of how there has been antigenic drift happening since the last year, and they create a vaccine based on that. That's why there's a new flu vaccine every year. The scientists basically take research on what is the current strains and what are the antigens that they have look like, and then they create a vaccine based on that. But when we have something like antigenic shift, this is when there is a sudden change occurring, and then the viral strain that can arise due to different strains that are recombining with each other. And that's when we are creating very dangerous strains, possibly even pandemic worthy strains, right? For example, due to the novel nature, newly created viral strains, via antigenic shift are highly infectious. For example, your 2009 swine flu pandemic arose due to the recombination of your classic swine virus and the avian influenza strain. When they recombine their DNA together, they suddenly emerge as the swine flu virus that is highly infectious and also dangerous. So in overall, antigenic shift is much more worse and dangerous compared to antigenic drift, right? This one is slow and gradual, accruing mutations to become a new subtype. But this is an instant cheat code that viruses can take to become a new subtype by just recombining the DNA with a new strain. Right? This is what you can, right? So this is when basically the avian and the human influenza strains recombine with each other with the classic swine to become the North American swine, and that recombined with each other to become the 2009 swine flu strain. Okay, so antigenic drift is slow and gradual. Antigenic shift is a very quick and very sudden and significant mutations that can occur due to the fact that the viruses are recombining through viral recombination. All right. So here's your summary.